Hip, hip, hooray for DNA. It provides the key to the plans for making everything in you and me.
Welcome to another past HSC exam question videos. In this video we're going to cover this exam question, which comes from the DNA to Protein chapter of the Blueprint of Life module. What I'll do in a second, I'll read the actual question. Once I read the question, you get about 5 seconds to pause the video. Once you pause the video, attempt the question, and then press play when you're ready. The question itself says the table shows that 20% of the bases in a section of double-stranded DNA are adenine. Complete the table below by identifying the other 3 base types and calculate the percentages of each base type in the section of double-stranded DNA. And B, construct a simple flowchart to describe the process of DNA replication. That's worth 3 marks. Alright, so when you're ready, pause the video and attempt the question. Welcome back. Alright, for the first, because all we have to do for the first one is we have to complete the table. And we should remember that A and T bind together and G and C bind together. So we know that if, for example, A is 20% for A, then there should also be 20% for T, because they're just, whenever there's an A, there's a T. The ratios are always the same. That was Chagaff's rule. So A is 20%, then T should also be 20%. And remember, we have U is not important for this. U is not important, uracil. And the reason why is because we're talking about DNA. In DNA, we only have 4 bases, and they are A, C, G, C. U is for RNA. So now that we've established this, so far, we get a 1 mark because it gets you 1 mark for every correct one. And how do we know how many G and C's there are? Well, overall, all we have for DNA are these 4 bases, A, T, G, C. So if we know A and T, they make up 20% each. That means 40% are gone. So the rest, these 2 must together make up 60%. These together make up 40%. To get to 100%, these make up 60%. And we know that, again, they're in a 1-to-1 ratio, so we just have to half this. So we know that G is 30% and C is 30%. And then we have our answer. So if you're a bit familiar with how this works, this would be probably the easiest or the fastest 3 marks you would have gotten in your exam, because yeah, it's just literally not much writing and just filling out a table, and that gets you 3 points. That's quite a bit. So 3 marks out of 3 for this. And the next one is construct a simple flowchart. And flowchart just means, you have start with A, go to B, go to C, go to D, etc. And then you can write on top of the arrow how it happens as well if you wanted to. So we have to construct a simple flowchart to describe the process of DNA replication. And that's worth 3 marks. So what you should start with, you've got your original DNA. You want to make sure you have it unwinding somewhere, so that unwinds. Then it gets copied. And then we have 2 new strands from those 1 original strand. That's the main parts you need to put in there. And you can also write the enzymes that were responsible for this as well. So what I wrote here is original DNA goes and unwinds, and the DNA unwinds. And the reason why it unwinds is because of DNA helicase, and you could have written that on top if you wanted to. So this DNA and helicase unwinds it. And then this DNA unwinds, and what happens next is these, after it's unwound, 3 nucleotides attach to match original base pairs. Right, so once it's unwound, then this DNA polymerase will come and make copies of it, of the unwound strands, complementary copies. And that obviously was done by the DNA polymerase. What happens next is, after all of this is done, there are 2 strands which are formed, right? So we've gone from 1 to 2 strands, and then they twist up again. So then DNA twists up into the original helical shape. So here they were a helical shape, like that. And then they've got unwound here. And at the end, they'll be formed, and then they'll unwind in their original shape. Only difference is now we have 2 as opposed to 1. That will get your marks. I don't know exactly for which statements you get your marks, but overall, for mentioning the correct sequence, for saying, we go from original DNA to DNA unwinding, to nucleotides being matched, to two strands being formed, to DNA twisting into helical shape, maybe also for mentioning the enzymes, that would get you your 3 marks. It might be that each of these correct parts is worth half a mark. Overall, that's your 3 marks out of 3. So you wouldn't have to write much because this is construct a simple flow chart. Where does it come from? It comes from this dot point. Describe the process of DNA replication and explain its significance. We didn't have to explain its significance, we only had to describe the process of DNA replication. I hope that was useful.
Thank you for watching.